So let us understand what IR spectroscopy is. We will take example of water molecule. So there are covalent bonds over here, and what we need to know is this, that covalent bonds are not static. They are in constant motion, and these are called vibrations or oscillations. So whenever we report a bond length, for example, the OH bond length in water molecule is 0.94 angstrom, we only report the average bond length because the molecule is in motion at all times. So this is only the average bond length at given. So there are two types of bond vibrations or oscillations: stretching vibrations and bending vibrations. In order to understand these, let us compare bond to a spring, a vibrating spring. So here we have a spring which is connected to two masses. So stretching vibration is when we hold these masses with our right and left hand and we pull them outward, and that is a stretching vibration. Then the bending vibrations are different kinds. There is a scissoring vibration where we hold the atoms with our thumb and index finger, and then we move them inward. So that is a scissoring motion. And then there are other kind of bending vibrations as well. So stretching a spring always takes more energy. Similarly, stretching a bond also takes more energy. And bending a spring or a bond requires relatively less energy. We will talk about this more a little bit later. So these stretching and bending vibrations of a covalent bond are associated with certain frequencies, and these frequencies fall in the IR region of the electromagnetic spectrum. So this is what happens in IR spectroscopy. So we have a instrument which is called IR spectrophotometer. It generates IR light. Sample is placed in the spectrophotometer and it is irradiated with IR light. So IR light consists of frequencies from 4000 centimeter inverse to 400 centimeter inverse. So the frequencies that match with the frequencies of the stretching and bending vibrations in the sample are absorbed, and a peak is observed in the IR spectrum corresponding to the absorbed frequency. So these absorbed frequencies or these peaks, they tell us about the types or functional groups present in the molecule. So this is how a typical IR spectrum looks like. On the x-axis we have frequency, and it is expressed in centimeter inverse. And then on the y-axis we have percentage transmittance, and basically percentage transmittance indicates the amount of light or the percentage of light that is transmitted through the sample, that travels through the sample without absorbance. And greater the value of percentage transmittance, the less the light is absorbed at that frequency. So for example, the percentage transmittance at 4000 centimeter inverse is 95%, and that indicates that the IR light of frequency 4000 centimeter inverse is not absorbed by this sample. And then at around 3400 centimeter inverse, the percentage transmittance is 10% or so, and that indicates that IR light of 3400 100 centimeter inverse frequency is greatly absorbed by the sample. Similarly, these transmittance for these peaks over here is very low, which indicates that IR light of frequency between 3000 centimeter inverse to 2800 centimeter inverse is absorbed by the sample. Notice that all of the light in this region of the IR spectrum is transmitted almost fully through this sample, so no absorbance occurs in this area, and then there are a lot of absorbances below 1500. So in IR spectroscopy, each of these peaks that we see represent a functional group or a bond. So for example, the peak at 3400 centimeter inverse is indicative of the presence of an alcohol group, an OH bond. Specifically, this peak is caused as a result of an OH stretch. Similarly, peaks between 3000 to 2800 centimeter inverse represent carbon-hydrogen stretches. And then there is a region below 1500 centimeter inverse that contains lot of peaks, so it is very messy, so it's not very useful. So for this class we're not going to worry about region below 1500 centimeter inverse. So this region of the IR spectrum below 1500 is called fingerprint region. It is called a fingerprint region because every compound has unique pattern of peaks in the fingerprint region of the IR spectrum, just like all of us have unique fingerprints. So the region above 1500 centimeter inverse is the most useful, and it is called the diagnostic region of the infrared spectrum. And that is where the functional group identification takes place. Now notice that the OH stretch appears at 3400 centimeter inverse, and the CH stretches appear at a lower frequency between 3000 to 2800 centimeter inverse. What is the reason for that? So let us look at that on the next slide.